The transesterification reactions are mediated by a huge molecular machine called the spliceosome. This complex comprises about 150 proteins and 5 RNAs and is similar in size to a ribosome. In performing even a single splicing reaction, the spliceosome hydrolyzes several molecules of ATP. So the assembling of this spliceosome needs energy. Strikingly, it is believed that many of the functions of the spliceosome are performed by its RNA components rather than the proteins. Thus, RNAs locate these sequence elements at the intron-exon borders and likely participate in the catalysis of the splicing reactions itself. So these are the RNAs which identify the sequences where the splicing should take place, and then catalysis, that is also controlled by these RNAs, and proteins have no any roles in these reactions. The 5 RNAs, that is U1, U2, U4, U5 and U6, are collectively called small nuclear RNAs or snRNAs. Each of these RNAs is between 100 to 300 nucleotides long in most eukaryotes and is complexed with several proteins. So several proteins are attached with these RNAs. These RNA protein complexes are called small nuclear ribonuclear proteins, snRNPs, or normally they are pronounced as SNRPs. This spliceosome is the large complex made up of these SNRPs, but the exact makeup differs at different stages of the splicing reaction. Different SNRPs come and go at different times, each performing particular function in the reaction. So this spliceosome is a dynamic complex. Some SNRPs come, they join, some SNRPs are living, so it is continuously changing under different conditions. There are also many proteins within the spliceosome that are not part of SNRPs, and others besides that are only loosely bound to the spliceosomes. The SNRPs have 3 main roles in splicing. First, they recognize the 5' splice site and the branch site. Second, they bring those sites together as required. And third, they catalyze the RNA cleavage and joining reactions. So these 3 are performed by these SNRPs. To perform these functions, RNA-RNA, RNA-protein and protein-protein interactions are all important. Let us consider some RNA-RNA interactions which play a role in these reactions. These interactions operate within individual SNRPs, between different SNRPs, and between SNRPs and the pre-mRNA. So these are different types of interactions. The figure below shows the interaction through complementary base pairing of the U1 snRNA and 5' splice site in the pre-mRNA. Subsequently, in the reaction, the splice site is recognized by U6 snRNA. So in this figure, you can see the U1. This U1 is attached to this sequence, and the attachment is through complementary base pairing. That is A, U, C, G, U, A, U, A, U, C, G. And similarly here, U6 is attached to this sequence. So in this way, these SNRPs and these RNAs are attached to the pre-mRNA. In another example shown below, the branch site is recognized by the U2 snRNA. A third example shows an interaction between U2 and U6 snRNAs. This brings the 5' splice site and the branch site together. So here you can see different interactions. This is between U6 and U2. This is between U2 and branch point sequence, and this is between BBP and U2, different pre-mRNAs. So these are the different interactions. It is these and other similar interactions and the rearrangements they lead to that drive the splicing reaction and contributes to its precision. That is the precision of splicing. Some known synapses are also involved in splicing. One example is U2AF, that is U2 auxiliary factor, recognizes the polypyrimidine PY tract, that is 3' spliced site. And then in the initial step of the splicing reaction, it helps another protein, branch point binding protein, BBP, bind to the branch site. BBP, also known as SF1, is then displaced by U2 synapse. So these are the different roles which are played by these snRNAs and synapses. Other proteins involved in the splicing reaction include RNA annealing factors, which help load synapses onto the mRNA, and DEAD or dead box helicase proteins. The latter use their ATPase activity to dissociate given RNA-RNA interactions, allowing alternative pairs to form and thereby driving the rearrangements that occur through the splicing reactions. They are also required to remove spliced mRNA from the spliceosome and trigger spliceosome disassembly. So once the RNA splicing has been done, so this spliced RNA must be removed, and this is removed through these actions, and then the spliceosome should also dismantle. So this is the spliceosome.